Welcome everybody to our webinar titled Perlecan Promotes Structural, Contractile and Metabolic Development of Human Cardiomyocytes. We're thrilled to have you join us for this enlightening session and for our first webinar. A few organizational points before we start. Please stay muted during the entire call. If you have any questions for the speaker, please submit them in the chat below, and we will discuss those questions in the Q&A at the end of the presentation. If any questions come up later on, we are happy to forward them to James, and I think he's happy to answer them too. Just reply to webinar@denovomatrix.com, and we will make sure that he gets those questions. As you've seen, this session is recorded, and it will be later available at denovomatrix.com. Before we dive into James' presentation, a few words from Dejan, CEO of denovoMatrix, on our technology and the biology behind our ECM. Dejan, please.
Thanks a lot, Sandra. Welcome to everybody from my side as well. So I'm going to briefly introduce you guys to sort of the sciencey part behind denovoMatrix. So denovoMatrix was founded on the idea that we need to create a very, very specific extracellular matrix for each cell type. And therefore, I thought to sort of give you guys a quick recap of the extracellular matrix and what are the various components incorporated in it. So as you can see from this diagram, the extracellular matrix actually has multiple types of molecules, including proteinaceous component, a glycosaminoglycan component as well. And then, of course, you have sort of the cell side. So that means membrane proteins, integrins, syndicants, and so on. And in order to recapitulate the extracellular matrix faithfully and in a way which is sort of maximizing a certain cell response or healthy growth, you want to recreate not just the proteinaceous part, but potentially also the sugar or glycosaminoglycan component. And as you might all be aware, most technologies on the market which are recreating the ECM are recombinant proteins or a mixture of recombinant proteins, things like laminin, fibronectin, vitronectin. But a lot of these solutions fail to recapitulate sort of this other part, which is equally important. So the glycosaminoglycan component, which is equally important and plays key roles in for example, growth factor maintenance or signaling. So what denovoMatrix has done, what is denovoMatrix all about? Essentially, we have created a coating technology that uses short peptide sequences, less than 30 amino acids long, to recapitulate multiple proteinaceous aspects of the extracellular matrix. In addition, we also incorporate sulfated glycosaminoglycans to mimic the glycosaminoglycan portion of the ECM. And what we essentially do is we offer a screening service, which looks a little bit like this. So it's a 96 well plate, which has a combination of 4 different glycosaminoglycans and 24 different biomimetic peptides, which mimic various extracellular matrix proteins, adhesion proteins, like I've mentioned, fibronectin, laminin, vitronectin, collagen, but also other diverse proteins found in the ECM, which could be, for example, cytokines. They could be cell-cell contact molecules like the cadherins, and so on. And using this platform, we've supported a variety of different customers, 120 and counting so far, with developing customized extracellular matrices that are specific to a certain cell type and are also chemically defined, and where we can learn something about what is important to these individual cell types. I would leave it at that and hand over then to Sandra again for introducing James and the exciting research that they've done.
Thank you, Dejan. So I haven't mentioned, but if you have any questions to Dejan, obviously you can also submit them in the chat. As I said, today we have the privilege of hosting Dr. James Smith, an esteemed researcher with a wealth of experience in cardiovascular biology and regenerative medicine. Dr. Smith completed his PhD at the University of Birmingham, where he delved into mesenchymal stem cells and the extracellular matrix. Following this, he pursued postdoctoral training at the University of Nottingham, focusing on automated manufacturing technologies for human pluripotent stem cells. His dedication to advancing cardiovascular science led him to be appointed as a senior research fellow at the BHF Regenerative Medicine Center, where he made significant contributions to the field. In 2019, Dr. Smith established his own research group at the University of East Anglia, where he's currently as well, leveraging cutting-edge techniques such as CRISPR gene editing and human-induced pluripotent stem cells to explore cardiovascular disease mechanisms. Today, Dr. James Smith will delve into his latest research endeavors, specifically investigating the role of Perlecan in cardiomyocyte development. His insight promised to shed light on crucial aspects of cardiac biology with implications for understanding and potentially treating cardiovascular diseases. Without further ado, I would like to welcome you, James, and please share your work and unravel the mysteries of Perlecan in cardiomyocyte development.
Thank you very much, Sandra. Can you see my slides?
Yes.
Are they sharing okay?
We can see your slides and we can see you. Go ahead.
Lovely. So thank you very much. Yes, so I'm going to be talking about our work investigating the role of Perlecan in the maturation of pluripotent stem cell derived cardiomyocytes. So start off with a bit of background about pluripotent stem cells and the cardiomyocytes that we make from them. So iPSCs are cells that we can reprogram from an individual with a particular genotype that we might want to study in a disease setting or from a healthy individual. So these cells have a lot of capacity and power. They can be differentiated into virtually any cell type of the adult human body. And the cell type that we work with in my group are the cardiomyocytes. So these are the contractile muscle cell of the heart, and they have several different applications. So in research, their power is in disease models, so trying to unravel the molecular mechanisms which underlie specific cardiac diseases. But they also have potential in the drug development pipeline. And their clinical use is starting to increase. So using these cells in patients for the potential treatment of things like heart failure or genetic cardiomyopathies. So if you look at the cell types that are currently being used from iPSCs, three main cell types dominate the market, and it's predicted to continue in that fashion. So these are the cardiomyocytes, the neurons, and the hepatocytes. And of these, you can see the cardiomyocytes have the dominant share of the market. And this is mostly due to their power in drug development initiatives. So looking for potential harmful cardiotoxic side effects, and also in the discovery of new drugs with therapeutic potential. As I say, their clinical uses are only just beginning, really, and as such, the market is predicted to increase and double over the next few years, and iPSC-derived cardiomyocytes are already in clinical trials for the treatment of conditions such as heart failure, myocardial ischemia, and dilated cardiomyopathy. So these are only predicted to increase, and therefore the use and potential of these cells is only predicted to increase further as well. So they're a useful cell type to be working with because of these applications, but unfortunately there's still a problem. So we've been able to differentiate these cells into cardiomyocytes for about 15, 20 years, but unfortunately we can't fully mature them. So the cardiomyocytes that we make from iPSCs still have an immature phenotype. So they're not exactly the same as the mature cardiomyocytes that you would find in the adult heart, which is a limitation across the stem cell field. So we've reviewed the differences between adult cardiomyocytes and stem cell derived cardiomyocytes. This is a few years old now, it's from 2016. And you can see there's a whole lot of differences across structure, gene expression, metabolism, ion channel expression, kinetics, etc. So again, this isn't ideal. And why it's not ideal is because we want the best cell types for any of these applications, whether it be disease modeling or drug testing or cell therapies. We want to make them as close to the adult human heart cells as possible. And to do that, we need to overcome this barrier of maturation, which currently we're not able to do. So again, here's another schematic kind of showing some of the differences between the immature and the mature cardiomyocytes. And you can see one of the key differences is the proliferative capacity. So the immature cardiomyocytes have a level of proliferative capacity, and as they mature, they lose this capacity and they become more structurally functional. Now, maturation is pushing them one way, but there's also a level of dedifferentiation which can occur. So in particular, heart disease settings, and this is different for different species, a level of dedifferentiation can occur, producing these more structurally immature cell types, which can lead to things like heart failure. So understanding this dynamic is important for both disease modeling, but also regenerative medicine. So as I say, here's a schematic of that dichotomy, with maturation pushing the cells one way, dedifferentiation pushing them the other way, and the switch between the proliferative and the non-proliferative cell type. So in the field of regenerative medicine, lots of people are studying the signals that are important for dedifferentiation. And when we look at the factors that they've found that can play a role, one particularly stood out to me, which was agrin. So agrin is a basement membrane heparin sulfate proteoglycan, and it was found to have remarkable regenerative capacity. You could take cardiomyocytes, expose them to agrin, and it would induce a level of dedifferentiation and proliferative capacity in these cardiomyocytes. So this was reported back in 2017 in this Nature paper, where they looked at P1 mouse hearts where the cardiomyocytes are proliferative, and they showed that agrin expression was high. And then as these switched postnatally to P7, the agrin expression was down-regulated. And they could visualize this as well with Chi67 positive cardiomyocytes. And again, show administering agrin could reactivate a level of proliferation in the cardiomyocytes. And the mechanism behind it was this proposed diagram here, where you have an extracellular matrix which is high in agrin. Agrin would bind the dystroglycan complex and allow a signaling cascade which would promote proliferation at the expense of maturation. And then what would happen is agrin would be replaced in the matrix by laminin or other ECM components which had the capacity to bind the dystroglycan complex through laminar domains, and this would switch off proliferation and promote maturation. So follow-on work from this kind of showed the regenerative capacity of recombinant agrin. So in this follow-on paper, the authors took a pig myocardial infarction model. So they basically induced a heart attack in pigs, and they showed that either a single dose of agrin or a double dose of agrin was sufficient to induce cardiomyocyte proliferation, which actually restored a level of functional capacity of the heart. So it was really exciting findings for the field. But there are unanswered and open questions, and some of them have been reviewed in particular review papers, such as the one shown here, talking about this agrin-mediated cardiac regeneration and what we understand and what we don't understand. And when you look at some of the open questions, it's about the dynamics about how agrin binding to the dystroglycan is controlled postnatally during maturation. And what's the role played by other ECM components? And do they have the capacity to outcompete agrin or have a higher affinity for the dystroglycan complex? Again, we don't fully understand this dynamic. And this is where our work on Perlecan comes in. So previously, plenty of people have shown that Perlecan is important for cardiac development. If you knock it out in mouse models, it can be embryonic lethal due to cardiac defects. But why we were particularly interested in it is because agrin and Perlecan are the two non-collagenous heparin sulfate proteoglycans of the basement membrane. And they're very structurally similar at the C terminus, with these 3 repeating laminar globular domains that have the capacity to bind the dystroglycan complex. In fact, Perlecan has been predicted to bind the dystroglycan complex with a greater affinity than agrin, which again drew our attention to its potential in this phenotypic switch during maturation, and to ask the question, does Perlecan play a role in the phenotypic switch of cardiomyocytes during maturation? So this is work carried out by my PhD student, Ben Johnson, who has graduated now and gone on to a fellowship. And Ben was using a differentiation protocol developed by Andrea Bernardo's group at Imperial College London, which can produce contractile cardiomyocytes after 6 days, but we subject them to a full 30-day protocol, which allows a level of maturation to occur. Obviously not the same level of maturation that would occur in vivo, but a level of maturation occurs. And we analyzed the day 6 proliferative cardiomyocytes to the day 30 cardiomyocytes, and we looked at the gene expression of Perlecan, and we did this through RNA sequencing of multiple cell lines and some targeted qPCR. And we saw, unlike agrin, there was a significant increase in the gene expression of Perlecan as the cells switched from the early day 6 to the later day 30 cardiomyocytes. We did unbiased proteomics, and again, between the day 6 and the day 30 cardiomyocytes, we saw a whole range of proteins which changed, as you'd expect, and one of the ones which significantly increased was indeed Perlecan, going up by a fold of about 3-fold. We confirmed this was happening in vivo, so we analyzed mouse hearts, again at the early proliferative points of P0 and P2 and the later non-proliferative points at P7 and P11, and again, saw an increase in Perlecan accumulation as the heart matured postnatally. So we were seeing that the expression of Perlecan on both a gene and protein level was increasing, but we wanted to know what its functional effects were. So for this, we utilized CRISPR gene editing. We produced a stem cell line which had 50% Perlecan expression. So we'd knocked out Perlecan expression from one allele, and we confirmed that expression across the multiple domains of Perlecan and showed that the iPSCs looked relatively unaffected. Their growth rate was the same, their expression of pluripotency markers was the same. So what would happen if we differentiated these lines into the cardiomyocytes? So we did this and we performed RNA-seq at multiple different time points. So we have our stem cells over here at day 0, and then our cardiomyocytes clustering from day 6 all the way down to day 30. And the genotype induced differentially expressed genes, you can see at all the time points there were a number of genes that were dysregulated by this knockdown of Perlecan. But significantly at day 30, this is where the greatest number of DEGs were seen, basically. So by knocking down Perlecan, the greatest effect was seen at the later maturation state of the cardiomyocytes. And when we clustered these genes at this final time point, again, they all clustered into processes that we'd probably associate with maturation. Things like cell differentiation, contraction, calcium handling, metabolic processes. Unsurprisingly, as Perlecan is one of the master regulators of the matrix, we saw a whole range of matrix-associated genes which were dysregulated by the Perlecan knockdown. So again, this was indicating that Perlecan's effect was seen at the highest point at the later maturation stage. And why this was even more interesting was because if you look at the differentially expressed genes between the time points, if you were to look between day 0 when they're stem cells and day 6 when they're cardiomyocytes, as you would expect, this is where you see the greatest number of differentially expressed genes from a time point of view, yet this is where we saw relatively few genes caused by the knockdown of Perlecan. So again, this was interesting to us because it supported the hypothesis that Perlecan was having its effect later on, but was there still something we could learn from these early time points about what Perlecan was affecting? So we compared the differentially expressed genes at these early points, so before day 20 and 30, and we saw 11 common genes that were dysregulated by the Perlecan knockdown. Reassuringly, one of them was Perlecan, which is what we'd done by the gene editing, and then the 10 others were potentially interesting targets that we can follow up on. So there's things associated with iron handling, metabolic processes, calcium handling. So again, there's some interesting potential effects that are seen even before the maturation stage. So what was happening at the later point? So if we took the day 30 cardiomyocytes and did the proteomics on the wild type and the Perlecan knockdown, again, we could see a whole range of proteins that were changed at this time point. Again, reassuringly, it confirmed the Perlecan knockdown, and then a whole load of other proteins that were knocked down or dysregulated as an effect. And they clustered into a whole range of processes. And one of the ones which really stood out to us was cell cycle progression. So as I've mentioned, there's this proliferative switch which occurs during maturation, and the proteomics data was pointing towards a change in the cell cycle progression caused by the Perlecan knockdown at this later time point. So we did Ki-67 staining to mark the proliferative nuclei within the cardiomyocyte population, and we saw in the wild type cells at day 30, there were relatively few proliferative cells in the population, but the Perlecan knockdown, there was significantly more. So this transition to the non-proliferative state seems to have been hampered by the knockdown of Perlecan, further supporting the signaling role of Perlecan in this maturation progression. One of the other processes that occurs during maturation is a switch in metabolism. So the cells switch from predominantly relying on glycolysis to more specialized fatty acid oxidation in the mature cardiomyocytes to meet the energy demands of the heart. So we did a range of metabolic analysis processes, including biolog mitoplate assays and seahorse analysis. And again, we saw that the day 30 cardiomyocytes had a significantly lower capacity in their ability to metabolize fatty acid substrates and a higher level of glycolytic reserve. So again, supporting the hypothesis that these Perlecan mutants were in a more immature state in terms of their metabolism. So we could promote maturation further. So all of the analysis I've shown so far was on 2D cultures, but we could encapsulate the cardiomyocytes into 3D engineered heart tissue 3D constructs, which are formed in well plate assays bound between two silicon posts. And we know the mechanical properties of these silicon posts. So as they deflect, we can calculate the force being produced by the cardiomyocytes, because we'd expect as maturation continues, their functional capacity and contractile function increases. Interestingly, even before we got to the contraction analysis, we could see a striking difference in the tissues that formed between the wild type and the isogenic Perlecan knockdowns, with these thinner tissues being produced, indicating potential reduction in hypertrophic growth or enhanced matrix remodeling had occurred. And we could quantify this and show these tissues were significantly thinner and were producing significantly less force. Again, further characteristics of immaturity. So all of the work so far has been investigating the role of Perlecan by using the genetic knockdown. But what we really wanted to do was see if we could take our wild type cells and enhance their maturation, potentially by growing them on Perlecan substrates, which is not an easy thing to do. Perlecan's a very large molecule. It's a proteoglycan. There's a lot of glycosylation which occurs. And therefore, buying full intact Perlecan recombinantly is not possible, unfortunately. So we're looking for alternative strategies, and this is where we came across the screen matrix from denovoMatrix. And again, one of the interesting things about the technology was you can see a whole range of peptides here, and then the attached GAG component, which you can have. And we saw that they were using a Perlecan peptide, either with or without the RGD peptide for enhancing adhesion. So we contacted denovoMatrix, and they gave us some details about the peptide. It was a C-terminus peptide, which again was interesting to us with our hypothesis with where Perlecan would bind. So we first obtained these screen matrices and seeded our cardiomyocytes to see if they would attach within these different plates. And again, we saw a whole range of different morphologies coming up, which was really interesting. There's probably a whole project in its own right investigating which of these peptides is best for inducing different phenotypes in the cardiomyocytes. But for this particular project, what we wanted to do was specifically focus on the Perlecan peptide. So de novo made some custom plates for us with a whole range of different concentrations of Perlecan, with and without the attached glycoaminoglycan, which we could use for our study. So what we did to start with is just use the Perlecan peptide by itself and switch it out for the final 10 days of maturation. So when we do this process normally, the final 10 days occurs on a vitronectin peptide, and therefore we wanted to see what would happen if we switched out our standard protocol to carrying out this 10 days maturation on a Perlecan peptide by itself. And we saw a striking difference in the cells that matured on these substrates. So you can see the vitronectin cells here. You can see they're stained for alpha-actinin, which is a cardiomyocyte marker in green. The nuclei is stained blue with DAPI. And you can see when the cells were matured on the Perlecan peptide, there was a huge change in morphology. You saw a whole range of clustering occurring. You saw the presence of these very large hypertrophic cells that were becoming present. And they'd gone through this excessive level of multinucleation. And when you zoom in, you can see this nucleation is occurring either in a ring or a horseshoe, or potentially as like a cluster of grapes. And you can see if you quantified this nucleation with high content image analysis, although the mononucleated cells stayed the majority of the population, there was a significant reduction on the Perlecan peptide because binuclear and multinucleator cells had increased. We could detach the cells from the substrate and put them through a flow cytometer in order to accurately calculate their size. And we could see a significant increase in the cell size of the cardiomyocytes that have been matured on the Perlecan peptide surface. We could also do some gene expression of the cells on the substrates, and again, doing a targeted analysis of many of the candidates we detected as being affected from the Perlecan knockdown. We could see the gene expression in many different processes, structural, metabolism, ECM, calcium, were changed by culture on these substrates. So why this is important is, as I say, the early cardiomyocytes go through hyperplastic growth in a proliferative phase, but following birth, they go through a hypertrophic growth stage where they increase in size and increase in nucleation. And these are two of the measurements we've seen by growing the cells on a Perlecan peptide. It seemed to induce hypertrophic growth and multinucleation. A big caveat is I'm not saying the cells have matured on the peptide surface. If anything, they look to have gone through pathological hypertrophy, but it's just really interesting to observe that two of the key processes during maturation, this Perlecan peptide seems to be able to have the capacity to enhance that signaling in our cardiomyocytes, albeit in a potentially uncontrolled manner. So we've published this work now, so if you want to read more about it, then look it up. But to summarize it, we can see the expression of Perlecan increases as the cells mature. When we knock down Perlecan, we can show it hinders development in metabolism, proliferation, contraction. And if we were to use Perlecan peptide substrates, we can promote processes such as hypertrophic growth and multinucleation, which would be important during maturation. So just a couple of papers just to say how our work fits in with some of the literature that's come out in the last few years. So this was a paper where the authors were doing a 15-day iPSC to cardiomyocyte differentiation protocol, and they were looking at secreted components from the cardiomyocytes. And they picked up changes similar to us. So they saw RNA and protein expression of Perlecan increased during their differentiation, which is what we saw. But interestingly, something which we hadn't looked at is they were looking at the conditioned media and detecting Perlecan in the conditioned media, and they saw they could only really pick this up at their final time point, which is really interesting, whether that's just because Perlecan had to get to a certain level to be detected, or whether there's something which cleaves Perlecan, which comes up, or a secreted form of Perlecan which is expressed. Again, that's an interesting observation. This is another interesting paper. So the authors were trying to develop a maturation substrate using a biological extracellular matrix, which they derived from perinatal stem cells. So they have a biological matrix, which they showed could induce a level of maturation in cardiomyocytes, and they could compare it to non-maturation biological matrices or commercial matrices such as Matrigel. And they did mass spec between the biological ones, and they looked for the differences in the components. And one of the differences they highlight in the discussion was a significant increase in the Perlecan content from 7.4% in the non-maturation matrix to 19.7% in the maturation matrix. It wasn't the only difference. There was things like an increase in fibronectin from about 36 to 52%. But again, it is interesting that this matrix, which looks to have quite power in maturing cardiomyocytes, has a high Perlecan content. This is probably my favorite one though. So in the same way that I've shown you in this study, we can produce 3D tissues of iPSC-derived cardiomyocytes in vitro. The authors here were looking at the effect of transplanting them in vivo for 12 weeks. And again, this is actually the best way of maturing these tissues. We can't mature them in vitro, so this is the best way of doing it. You put it back in vivo and they go through an accelerated level of maturation. And they highlighted some differences between the in vitro and the in vivo more matured tissues. And again, they saw some differences in things like fibronectin, collagen 4, but the difference in Perlecan is really striking. You can see in the in vitro tissues, they can hardly detect Perlecan in green, but following 12 weeks of in vivo maturation, Perlecan goes through the roof. So again, this further supports our hypothesis that it's important in the process. And why that's important is because lots of people are looking at the strategies to produce these more mature cardiomyocytes. They're using things like the in vivo maturation, the 3D culture, or changing the ECM substrates. And again, if we can understand what it is about these processes that promote maturation, then we can probably enhance it further. And we highly are confident that Perlecan is playing a role in the switch towards this direction. And there's a further implication in therapeutic applications in the same way that the authors of the AGRIN study showed there was therapeutic potential of using recombinant agrin to increase the proliferation of these immature cells. In heart failure, you'd still want to revert these immature cells back to a more mature state. And potentially, if we can get to the bottom of Perlecan's signaling role, we might be able to come up with therapeutics that can enhance the maturation in vivo. So some acknowledgements, as I say, Ben Johnson, who is the first author on the paper, did the majority of the work. He's gone on to a Marie Curie Fellowship in the Netherlands, but collaborators at UEA, Linda and Stuart, Nottingham, Kathy Merry, Chris Dennin, Liverpool, Mandy Peffers, over in Australia, John Whitelock was a great help, and also at Imperial College London, Andrea Bernardo's techniques were essential for this study. And just very finally, if you're interested in proteoglycans such as Perlecan, the premier conference in the proteoglycan field is the Gordon Research Conference, which is happening later this year. I'll be presenting the Perlecan work and showing some unpublished data there. So if you're interested in hearing more about the story, then definitely check out this conference. If you're a student looking for a PhD, there's still a week left to apply for a PhD project that's being advertised in my lab, which is looking at the role of the extracellular matrix in cardiac development. And again, it's following on from the work I've presented here. And I will finish there, and I'm happy to answer any questions.
Thank you so much, James. I encourage all the listeners to put down your question into the chat. We received a couple of questions up front, and there's already one question in the chat. But I would probably start at the beginning when you mentioned in your presentation that Perlecan has like overlap with agrin, and you knew already that agrin plays an important role in cardiomyocyte biology. Was there any other proteins that you have tested at that point, or were you always targeted onto Perlecan?
Yeah, so the project was had a particular focus on Perlecan. So I don't think Perlecan is the only thing that plays a role. There's been a really good paper that's come out from Sanjay Sinha's lab at Cambridge looking at the role of fibronectin. But things like fibronectin and Perlecan interact within the matrix. You know, the reason why it's a matrix is because it's complex, and these things can activate multiple signaling pathways by working together. So Perlecan is one of the master regulators. So people have shown that when you disrupt Perlecan, you disrupt a large number of the pathways associated with matrix assembly. So again, it makes sense that the pathways that it's activating are crucial pathways, and if you disrupt it, you're going to disrupt those pathways. But that's not to say that there aren't other parts of that complex in the matrix that you can't disrupt as well. So yeah, how we got onto it was John Whitelock is a professor in Australia who's been studying Perlecan for over 30 years. And he did a visiting professorship to the University of Nottingham, where I was based before. And the agrin paper had come out at the time, and me and him were discussing it, and tapping into his wealth of knowledge, we came up with this hypothesis and come up with a project to investigate it.
Okay. And another question that we have received is, what do you expect, or can you think of other ECM proteins that have a similar influence? You just mentioned a little bit that you were expecting lots of proteins from the ECM to do something to cardiomyocyte biology, but have you tapped into this? Can you do some speculation on that?
So we've not tapped into it too much, but I mean, in terms of the hypothesis about the dystroglycan complex, you're looking for components with laminin domains that have the capacity to bind the dystroglycan complex. So obviously, all the forms of laminin have these domains, but there's different forms of laminin. So potentially there's a whole lot of investigation that could be done looking at the different forms of laminin and how these can interact with the dystroglycan complex in different manners. But they're not the only proteins that have these laminar globular domains. So yeah, there's a whole spectrum of targets that could be tapped into, but yeah, that's not something we've done yet.
Yeah, maybe some proximity labeling in the mature cardiomyocytes might be interesting. Okay, so I'm still encouraging people to submit questions. We also received the question, which part of the Perlecan protein was used as the Perlecan peptide in the experiments? And I know you already mentioned in in the presentation that it was near the C terminus. Is that correct?
Yeah.
Yeah.
Yeah. And I mean, that's a valid point because there's a whole, that's one peptide and it showed remarkable functional effect. We were shocked. I'd never seen cells look like they did when we grew it on that peptide. But, there's a whole study there looking at different forms of these peptides. Perlecan is a multi-domained proteoglycan. It's got 5 domains, attached GAG components, which can influence signaling. So, yeah, probably if we're going to get to the bottom of the functional capacity of Perlecan, it won't be done by single peptides. But again, for this particular study, showing the capacity of a C-terminus peptide to induce those processes was really important for us.
Is there any mammalian system or a cell line that can produce full-length Perlecan peptide that you can actually isolate and use?
Potentially. I think this is where you get into the complexities of cell type specific isoforms. So, you could use a human cell type and you have the advantage that the glycosylations would be human, etc. But again, it's still there's questions about whether it's a relevant cell type. I think there might have been a question in the chat about the source of Perlecan.
Yeah.
And again, I think that's a really valid point that we have done some work looking at Perlecan isoforms and the differences between the forms produced by the cardiomyocytes and produced by the cardiac fibroblasts, which would be the supporting cells that would often be responsible for the majority of the matrix that the cardiomyocytes would see. And again, there are differences in isoform expression and forms. So again, it's going to be a very complex story to get to the bottom of. So yeah.
Is the glycosylation involved in the binding to the dystrophin complex?
Yeah, really interesting questions. There's some really good review papers on how glycosylation affects binding to the dystroglycan complex. So again, there's work to be done there. There's a great study by Rebecca Gundry's lab in the United States where they looked at glycosylation changes during cardiomyocyte maturation, and they saw 14 N-glycosylated peptides that increase with maturation, and 2 of those 14 were Perlecan glycosylated peptides. So again, I didn't mention that
Interesting.
study. Yeah, I didn't mention that study because it gets into the glycosylation side, which is a whole different story. But again, really, another paper which kind of supports our theory that it's important and playing a role.
Yeah. And have you tried to use the peptide to rescue the knockdown phenotype or the dysregulation of the metabolic pathway or something like that?
Yes and no. We did that experiment because it's a valid question. It's something we wanted to ask ourselves. But to be honest, by the time we got to working with the de novo peptides was right at the end of Ben's PhD. So he did a whole range of experiments, including the one you've described. And I think he isolated samples and they're probably sat in a freezer somewhere. So as I say, we're recruiting for a new PhD student who will continue some of this work. And again, there's potential experiments to kind of further interrogate rescue or rescue's interesting, but we want to go further. We want to take the wild type and push that further. It's kind of, we're confident that the mutant is immature. What we want to do is push it forward, and as you say, one of the ways of doing that would be to try and rescue the mutant and find peptides or combinations that can rescue the mutant and then use them on the wild type to push forward. But yeah, we've got some different strategies in mind as well.
What do you mean by pushing forward the wild type? Want to increase the concentration?
No, increase the maturation. So the whole problem with the field is the wild type cells that we mature at the moment aren't fully mature. They're not the same as in the adult human heart. So what we want to do is find a way of pushing them past the point we get them at the moment. And I believe that is possible if we can find the right way of exploiting the right kind of matrix signaling.
Yeah, speaking of that, actually, we also received the question, what's the influence of the GAG components from the denovoWebinar? And I think you mentioned you want to push further. Maybe that's a route to go.
Yeah. So the data I showed here was without the GAG component, but it's a valid question and one that probably deserves its own study in its own right to really interrogate what the effect of the different GAGs can have on promoting maturation. So that's kind of where the work's going, if I'm being honest.
Is there any knowledge what's the in vivo situation? What's the glycosaminoglycan that's most present in the heart?
Yeah, so there is some work about what changes with aging and disease. It's not so much on maturation, but kind of later stage aging. So things like heparan sulfate, the level of sulfation increases with aging. There's whole changes of sulfation patterns which can occur in different disease states that have been reported. But yeah, in terms of interrogating the mechanism in maturation, that's kind of something we're interested in doing. In fact, it's something we've started doing, but
Yeah.
we can't talk about that data too much, but we come to the
Okay.
proteoglycan Gordon conference.
But that brings a whole new level of complexity into the problem that you have also the sulfation of the GAGs, and to what degree do you need that?
Absolutely, yeah.
Yeah.
I couldn't agree more.
Yeah. I had a question. The Perlecan itself, when you completely knock it out, it's lethal for the cells, right? Because I was quite stunned that it doesn't do much to pluripotent stem cells, but has such a tremendous effect
Well,
later on.
I suppose that's I was maybe misleading with the data there, because what we've done is create the heterozygous knockdown, and the data I showed there showed the heterozygous knockdown of Perlecan had minimum effect on the stem cells, which don't express Perlecan at the same level that the mature cardiomyocytes do. But the other way of interpreting the data is we were unable to make a homozygous knockout. So that's clearly a limitation of this study,
Yeah.
and potentially the reason why we were unable to do it was because when we achieved it, the cells died. So we screened screened hundreds of clones and made heterozygous knockdowns in 3 different methods, multiple clones each time. And normally when you do that, you obviously would have, less homozygotes. It should be like 25% or lower, but you would find them at some
Yeah.
point, especially with the different strategies we use. But we screened hundreds of clones and never found one, which is indicating that potentially every time the dual editing occurred, it was killing the iPSCs. So yeah, saying that the heterozygous seemed relatively unaffected, and therefore Perlecan has no effect when they're in the stem cell state, isn't really fair, because potentially making the homozygous out was lethal. So it's clearly having an effect. It's known FGF signaling is crucial for pluripotent stem cells. So if you grow your pluripotent stem cells in defined media like E8, it's got extreme levels of FGF in it. That's the key growth factor. There's 2 growth factors, TGF, FGF, and FGF's at the much higher concentration. FGF is known to bind to the core Perlecan protein, and it's been shown to have an effect on stem cell in neural stem cells anyway. Perlecan FGF binding has been shown to be crucial for cell proliferation. So potentially by knocking Perlecan out fully, we were disrupting the FGF signaling, which is crucial for pluripotent stem cell expansion, and that's why every time we did it, the clone would die. So yeah, slightly misleading to say Perlecan is not essential in the stem cell state. It's more the fact that the heterozygous wasn't detrimental in the stem cell state.
Do you know whether there's any inducible knockout model of?
Yeah, you sound like the reviewer.
Sorry.
Yeah, yes, we looked into it. We couldn't find one, and again, it's complex for what we wanted to do, because you start to ask questions about when's the right time to induce. If you've got the cells there and they've been down for 15 days, they've laid down a whole matrix, and then if you induce the change there, you'd still have Perlecan there. If that makes sense, it would be a less clean system. You will have knocked it down because you won't keep making it, but it will still be there. So again, it's there's definitely studies that can be done with inducible systems, but yeah, we couldn't find a clean one for what we wanted to do.
Okay, well, that answers definitely. I think you have answered the first question in this chat. Another question.
Sandra,
What
may I
does?
come in? May I come in with one quick question for
Yeah,
James,
sure.
because it's on the topic. You mentioned also the sort of with the Perlecan peptide from the screen matrix that you got a certain change in the morphology and in the size of the cells that was like pathological. Is there something you can share in an insight as to what the ECM looks like when it's pathological for cardiomyocytes?
Well, that's a good question. So again, I've not shown, when I was talking about putting the work into context, there's another paper which sometimes I talk about where they induce hypertrophy of the cardiomyocyte. They use a chemical called endothelin, which induces kind of pathological hypertrophy. And they They did that on iPSC-derived cardiomyocytes, and then they did analysis of the matrix at different points after inducing hypertrophy, and they saw proteins that changed after 24 hours, 48, and 72. And I think there was 20 proteins in all that changed, and one of them which changed at the later time point was Perlecan. So again, potentially an increase in Perlecan does occur during the pathological hypertrophy in cardiomyocytes. In terms of in the heart, in terms of in vivo analysis of the cardiac matrix in disease, again, there's lots of work which is in different disease states and how it changes. Lots of work's been done on the mechanical properties, which is something I've not gone into too much here, but there seems to be some theories about agrin's role in regeneration and how it produces a relatively soft extracellular matrix, which supports proliferation. And then the matrix stiffens during development and can further stiffen during disease. And why that's potentially interesting to this work, if you remember the slide I showed, which showed the structure of agrin and the structure of Perlecan, Perlecan's much, much longer and is associated with a much stiffer matrix. So potentially, as well as changes in growth factor signaling and receptor engagement, there could be mechanical changes that are caused by the change in Perlecan. Obviously, that wouldn't be investigated in the same manner with single peptides.
Right. We're running a bit out of time, but I definitely want to address the questions in the chat. So what does induce the Perlecan expression? I think
What
that's mentioned, yeah, over time, I assume, doing that.
So we, that's a great question. There are papers out there which look at that. We didn't dig into that side of the mechanism, but there is work out there on what the promoters of Perlecan is and what can activate it. But again, that's not, we didn't end up having to dig into that side of it too much.
Okay. Did you do electrophysiological measurements to reveal maturation?
Yeah, great question. So there's lots of different ways of measuring maturation. So I've talked about some here, structural, metabolism, contraction, but electrophys was on the list, but we didn't. We did some calcium handling assays with calcium reporters and saw some really interesting differences in that. But again, we kind of ran out of time and it didn't go into the paper, but we wanted to do electrophysiology as well, but we didn't.
All right.
But again, it's interesting because there's some papers on Perlecan's effect in other cell systems, which show it can actually affect the opening of different ion channels, and therefore we would predict that there'd be a change in the electrophys. But yeah, we needed to find someone we don't have electrophys for cardiomyocytes set up in my lab at the moment, so we would have had to find a collaborator who was willing to do a lot of patch clamp, and yeah, we didn't do that in the end.
Is Perlecan expression involved in remodeling in other cardiac diseases?
Yeah, so as I say, there's work that's out there which has shown that when you induce disease states like hypertrophy, Perlecan does change. So again, there's definitely evidence out there. I think traditionally where Perlecan's been studied is angiogenesis. There's a really interesting dichotomy in how Perlecan affects angiogenesis. So I've mentioned the C-terminus of Perlecan. That actually can be cleaved, and when it's cleaved, it creates a peptide in its own right called endorepellin, and they show that full Perlecan has the opposing effects to endorepellin. So one is pro-angiogenic and the other is anti-angiogenic, and the cleaving of Perlecan can be really crucial in angiogenesis in different disease states. So again, slightly different to what I've talked about here, but traditionally that's where most of the research on Perlecan has focused.
Oh, okay, interesting. We're running out of time. You have mentioned a little bit where your work is heading towards the GAGs and investigating the role. Is there anything else you would like to share before we close?
No, I think that's covered most of it nicely. So yeah, just thank you again for the opportunity, and yeah, really enjoyed it.
Thank you very much, James. So apply for a PhD position in James' lab. He's a very nice guy. And thank you for listening, everybody. As I said, this presentation will be available on denovomatrix.com, and I wish you all a pleasant evening. Goodbye.